Welcome to today's presentation entitled OneLab, Enabling Connected Lab Automation and Fully Traceable Workflows. The COVID-19 pandemic has resulted in a challenging work environment, with organizations having to socially distance their workforces and many working remotely. This has made the development and commercialization of vaccines, treatments, and critically needed PCR-based and serological diagnostics especially challenging. The need for highly versatile, easy-to-use automation for the bench that delivers superior workflow reproducibility and productivity has never been higher. However, reproducibility has been an issue for many years. Let's consider this from a user standpoint. I have multiple complex workflows that need to deliver critical data systematically of absolute value and quality. This data quality is critical towards regulators and to build key decisions determining our success. Despite extensive training that took 6 months for each employee, the complexity of these new workflows is significant, and we keep seeing errors, inaccuracies, biases, mistakes, and differences in performance over time, or by different people across different sites. And this is because the data generating instruments are fantastically sophisticated, but the sample prep is complex and changes all the time. Every technician works slightly differently, despite extended training, and each day we're never sure of the outcome. Formal SOPs are as detailed as possible, with many tens of pages for each process, but nobody reads them, and too much space for different ways of doing things. Even myself, I don't know if I would actually be able to. So how does this make me feel? Well, despite everybody's hard work, actual data is not of the quality standard we achieve in ideal conditions, and often miss the required target. I'm not productive, and I know we need to change our approach. The problem of reproducibility in the life sciences is substantiated in the peer-reviewed literature and has been the case over many years now. Indeed, it's considered that nearly half of all preclinical research that is irreproducible costs over 28 billion, a huge sum of money. So savings here could be incredibly important in accelerating research and improving productivity. So why do we have these problems now? Well, we can consider this from different viewpoints. Let's consider the science first. Life science understanding has progressed exponentially, driving scientists towards more quantitative and critical data-generating workflows. Consider that the Nobel Prize for DNA discovery was awarded in 1962. In terms of experimental impact, the number of steps of a typical sample prep workflow has exponentially increased, including both heterogeneity and variability as well. For example, the BioNTech vaccine, 50,000 steps, but easy to modify. Incredible. Data generated from an experiment increased from 1 bit to 2.1 terabits, increasing the information value of correct and reproducible experimental procedures. Indeed, think about NGS pooling. From a human organizational standpoint, larger and larger groups working in collaboration, augmenting the reliance on external data without redundant repetitions for independent validation. Scale-up needs have changed, with personalized immunotherapies calling for scaling up of parallel workflows with intrinsic QC/QA for smoother research to manufacturing transition. If we consider the tools versus analytical instrumentation, the tools and methods assisting humans on experimental execution have remained substantially unchanged or progressed at a much slower pace. Consider the pipette. We saw the pipette in 1957 and the liquid handling workstation in the early to mid-80s. The conclusion of all of this is that the actual occurrence and cost of biases, errors, variability of workflows has increased. But we do know the sources of error. Indeed, there's been much published literature on this too, especially in recent years. And those sources of error include data dredging, omitting certain data, underpowered studies, technical errors associated with the workhorse tools of the laboratory, underspecified methods, and weak experimental design. From this, a number of potential solutions have been identified. the need for more open data, registered methods, collaboration, tools for collaboration, greater degree of automation, open methods, reporting guidelines, and post-publication review. But we also have to look at this in terms of reproducibility among what and among who. And there are a number of use cases that are important here. Between assay provider and end user, between scientist and scientist, between scientist and technician, in programs involving multiple scientists and technician, for example, in ring trials, from research to manufacturing. Ideally, an automation platform should support all the use cases above. With that said, enter the connected lab. Andrew Alliance automation products are centered around OneLab, the connected lab and automation software. OneLab is a unique browser-based software environment that runs on any tablet or PC. It provides an intuitive graphical interface to create device-independent protocols and complete workflows. A single protocol designed by a scientist can be carried out fully automated with an Andrew Plus, guided with Pipette Plus and connected devices, or with non-connected pipettes and lab products. Notably, users can also benefit from an online library for rapid access of pre-validated automation scripts, and the software is now CFR 21 Part 11 compliant. OneLab also enables full sample tracking and sample data import-export, and is fully Labware vendor agnostic. Andrew Alliance's automation products feature an award-winning pipette robot, Andrew Plus, and an award-winning guided pipetting system called Pipette Plus. Both are remotely programmed by OneLab, which carries the additional advantage of full traceability and seamless method transfer as a benefit of using the cloud. Andrew Plus hosts a versatile modular deck whose Domino Labware holders are compatible with a wide range of labware, and whose connected ontologies such as shaking, heating, cooling, magnetic bead separation, and vacuum, together with connected column and plate gripper tools, support a wide range of applications. Best of all, its design means that capabilities can be readily added, essentially future-proofing the user. Pipette Plus provides remotely programmable electronic pipettes based on the market-leading Picus pipette from Sartorius, with the important benefits of reducing skills gap between novice and experienced user, as well as significantly reducing the time required to complete laborious manual pipetting operations, such as concentration normalizations and serial dilutions, typically by at least 80%. Imagine the productivity improvements there. Pipette Plus is currently being used in HG Pharma's mobile COVID-19 testing trucks in Europe. These cloud benefits come with a number of important perks, if you like, as we have seen during the course of COVID-19, in the words of James Broughton, research leader at Mammoth Biosciences on the west coast of the US.
One of the things that, we struggled with is some of our team members are working primarily from home, or not able to come into the lab every day. And so we were trying to figure out ways that we could really, keep the lab moving and keep them, those team members engaged with the science that's going on and keep everything going. And the Andrew Plus really allowed us to do that by, one, the software is, cloud-based. So we were able to kind of design experiments remotely. And then, Carly was able to work with another research associate in the lab and to kind of set up experiments on the Andrew Plus and then execute them remotely. And so we actually had set up a webcam so that Carly could watch the equipment run and be able to stop it if anything's going wrong, so that other people can keep doing their experiments and not have to keep an eye on the equipment.
OneLab is a browser-based software that can be accessed for free at onelab.andrewalliance.com. The software enables easy method development, traceability sharing of protocols, and workflow automation, all within one application. The most prominent buttons in OneLab allow you to quickly access the protocol designer to create new laboratory sample prep protocols. If you already have your protocols designed, simply click the Execute Experiment button to begin preparing samples. The Quick Access menu provides an overview of frequently used workspaces, which are collections of protocols based on user profiles or projects. Lower on the page, we can see Review, Recent Activity in the lab to see what protocols and experiments have been carried out and by whom. In the My Experiments panel, you can see a summary of your most recent experiments, as well as any ongoing experiments currently being executed in your connected lab. In the My Experiments panel, you can see a summary of your most recent experiments, as well as any On the bottom right side of the application window is a blue button leading to Intercom, the web-based service system. Click for assistance from our team of workflow and automation experts for real-time support. Now that we've seen a quick introduction to OneLab, let's see how it can be used for an LC-MS application. Reagent and plate preparation for LC-MS workflows can be laborious and time-consuming. Generating accurate calibration curves requires excellent pipetting technique, as well as adhering to best practice for good performance of quantitative applications. OneLab and its ecosystem of connected devices can improve assay accuracy and increase workflow efficiency in the lab. Nitrosamine impurity testing has increased dramatically in recent years because of its detection in a number of on-market drugs and because of the carcinogenic risk they pose. Drug developers and manufacturers are now obligated to test their incoming APIs and outgoing drug products for nitrosamine impurities, which is being done by either LC or LCMS. Our goal is to create a standard curve for quantitation of nitrosamines in drug substance and drug products. Here you can see an overview of our nitrosamines plate setup workspace in OneLab. Clicking on any consumable will bring up information about the reagent that consumable contains. Here we can see our microtube contains a nitrosamine 6 mix standard, denoted by the yellow color. Clicking on the info button, we can see the total volume of reagent, 40 microliters, as well as the reagent concentration, 250 micrograms per milliliter. Designing our liquid handling protocol is easy with OneLab. Each protocol step corresponds to an action, in this case, a pipetting action. In step 1, the nitrosamine 6 mix standard is dispensed into a microtube for dilution. To program this action, simply drag from your source to your destination and input the volume transfer desired, as well as any liquid handling or mixing settings. The next step of this protocol involves diluting the standard with 960 microliters of water from another consumable, in this case a Falcon tube, in the workspace. This action is again achieved via drag and drop from the source to the destination. After dilution, we transfer this standard into position H12 in the 96-well plate we will use for injection. To create standard in both the drug substance and drug product matrices, we perform the same pipetting steps with new reagent sources. In H11, we dispense standard diluted with drug substance, and in H10, standard diluted with drug product. Now let's see how we can confirm the concentrations of the diluted standard in each well of the 96-well plate to ensure no errors were made. Clicking on any colored well or consumable in OneLab allows us the opportunity to inspect the well to view the reagent definition at any point in the protocol. After transferring the diluted standards into the 96 well plate, we can click on each colored well to confirm our reagent concentration. Here we can see a view of each reagent definition, verifying the 1 microgram per milliliter concentration. The next step in the protocol is to generate a calibration curve. This involves multiple pipetting and mixing steps, ensuring that we start at the higher end of the curve range and avoid carryover on pipette tips. Additionally, we will include the transfer of 4 well blanks as per our plate map design. We repeat this process of generating calibration curves for the standard 6 mix diluted in drug substance and drug product, along with their corresponding blank wells. Here, we can see the plate map with rows C and D associated to the drug substance curve, and rows E and F for the drug product curve. Now that our protocol is programmed in OneLab, we can use the additional features in OneLab to help us execute our protocol. To start, we can use our standard lab pipettes manually and have OneLab provide step-by-step instructions for the sample preparation protocol. OneLab can also provide a time estimation to complete the entire protocol based on average user setting up and carrying out all over the pipetting and mixing steps. In this case, the estimation to generate this plate is about 1 hour. Choosing manual execution, OneLab will begin its pre-protocol check to ensure that we have what we need. This is the pre-protocol checklist to ensure the user has all of the materials required to carry out the protocol. OneLab even prescribes the number of tips that will be needed, as well as all of the consumables, reagents, and their starting concentrations. Confirming the starting materials will take us to the protocol execution guide, and this guide provides a visual aid of each pipetting step, with on-screen visuals for pipetting volume, source and destination, liquid level guidance, pipette technique, as well as any other comments from the protocol designer. This screen follows you along the entire execution journey, and at the end provides a summary report, which is stored in OneLab. I strongly recommend this further reading. This paper was published in SLAS Technology last November, and it talks about the highly versatile cloud-based automation solution for the remote design and execution of experiment protocols during the COVID-19 pandemic. And it speaks to some of the points that I've covered in this presentation with regards to both OneLab and the Andrew Plus pipetting robot. Some additional useful resources that I strongly encourage you to look at. There is general product information, both at andrewalliance.com and at waters.com/automate, especially application notes that are more LC-LCMS specific, like, for example, the application that I mentioned during the course of this presentation. Also, you can try the software and register for a free account at onelab.andrewalliance.com. If you have any questions related to compliance, we have an excellent traceability blog with articles written by our in-house expert, Heather Longdon, at blog.waters.com. And then also an excellent series of videos called the Simplify series that take you through OneLab, starting, if you like, with a simple navigation of OneLab itself, a bit of a tour, if you like, to also examples as to how to set up protocols like serial dilutions, concentration normalizations, and how to use the OneLab library.